Good morning, good afternoon, and good evening to all the participants in the third session of the acute flaximyelitis, what we have learned in order to be prepared. Thank you very much for being available for this virtual symposium. Today is exciting. This is an amazing session today dedicated to discuss issues related with pathogenesis, virology, and immunology of acute flaximyelitis. Next slide. So what we always like to emphasize is, next slide, is that we like to be very thankful and grateful with people that have contributed to the organization of this acute flaximyelitis symposium, the Johns Hopkins Myelitis Center, the Kennedy Krieger Institute, and particularly the acute flaximyelitis working group, that is a multi-center consortium of people, healthcare providers, families, and all people interested in acute flaximyelitis. We have great support from the SRNA, the Bar-MacLane Fund for Neuroimmunological Research, and our colleagues at the Children's National in DC. Next slide. So one thing that is extremely important is the SRNA is the Siegel Rare Neuroimmune Association, has contributed extensively to the organization and logistics of the acute flaximyelitis working group, as well as these virtual symposiums, and we are very grateful for their collaboration and support, and particularly to Roberta and Andrea, who have put a lot of effort in the organization of this virtual symposium. Today is a day dedicated to basic science, immunology, virology, and what I call translational research. And the acute flaximyelitis working group is comprised not only by clinicians, healthcare providers, but scientists and researchers that in different areas of the country that are highlighted here in this map are participating in the most amazing way to understand acute flaximyelitis. And I'd like to emphasize that the role of scientists, virologists, immunologists, and laboratory scientists in understanding this disorder, because every biological sample that is being analyzed by them is going to contribute extensively to understand this disorder, and in the future to generate data that allow us to better treatments and management. And you are going to hear from some of them, including our colleagues at NIH who are working in vaccines, how this amount of information from the virology and immunology group is contributing to the support of our research. I will pass my microphone to Cristina. Cristina, next slide.
Sure, so welcome back. This is session number 3. By now, probably we're all very adept at navigating in between the stage and the sessions. Today, we have 9 exciting presentations that are looking at the virus, the effect of the virus on the cell, on the organ and organism, the reaction, the immunity, and the genetics that are predisposing to different manifestations. And at the end, we're putting it all together with the help of NIH, looking at vaccines and future possible treatment, looking at immunity. And we're going to have a back and forth in between basic science and the clinical, and right back by Dr. Yen. Now, as you remember, the previous symposiums are available on the SRNA website. And so I am again inviting you to go there and digest each one of them at your own pace. Next week, we're going to move it to the long-term consequences and management of these consequences. So it's again, we're jumping back to the clinical stage. Then we're going to take a break of 1 week so we can all celebrate the 4th of July in the United States, and people that are not in the United States can take a week of a break from seeing our faces. And then we'll be finishing it on the 10th of July with very, very pertinent information about clinical interventions that could help improve function. Thank you for being here. Carlos, back to you.
Great, thank you so much. And let me introduce the lectures today. And the first is going to be a quite important lecture on looking on pathogenesis, mostly centered on the virus and what we have learned about the virus and the immune system. And that lecture is going to be given by Dr. Richard Sherman, who is at the Venter Institute at the University of California, San Diego. And the amount of data that is available already on enterovirus is quite amazing. And Richard is going to help us to understand that. The next lecture is going to be a very important lecture. And next slide, Roberta. The next lecture is going to be looking at how the virus, the suspected virus like Enterovirus D68, is affecting different cell populations in vitro models. And Dr. William Jackson at the University of Maryland is going to facilitate understanding that process. And if we go to the next slide, there is going to be a very important concept that we are going to be discussing is the concept of neurotropism of Enterovirus D68. And Dr. Amy Rosenfeld, who is at Columbia University in New York, is going to provide that information. And she has a quite fascinating model that will be very helpful for us for understanding how Enterovirus D68 is associated with acute flaccid myelitis. And after Dr. Rosenfeld, we are going to have Dr. Ken Tyler, who is at University of Colorado. And Ken is a neurologist, a basic scientist as well, and virologist, who is going to give us a very important view about animal models for studying AFM. And there is already a very amazing information about that. At the end of this lecture, we are going to move next to the panel discussion of these different topics. And it's going to take 20 minutes. Please send us all the questions in the chat. And we hope that we can have a very good discussion about this. Following the panel, we are going to move very quickly next to a very fascinating topic is what is the future of diagnostic tools and laboratory tools in acute flaccid myelitis. And Dr. Charlie Chu at UCSF, who already gave us a very nice overview of the virology and the first visual session, is going to provide that lecture. And that is going to be followed next by a fascinating topic is that the genetic susceptibility that patients may have to acute flaccid myelitis. And that is going to be given by Dr. Priya Dugal, who in the past several years have focused a tremendous amount of effort for understanding this concept of genetic susceptibility. And follow Dr. Dugal, we are going next to another very important topic is the topic of vaccines for enteroviruses and prevention of acute flaccid myelitis. And is going to be a very helpful to have Dr. Cristina Cassetti, who is a deputy director of NIAID, who is going to provide the view of what and how the NIH is working on developing vaccines for enterovirus. And this is going to be a very important lecture as well. And then we are going to move to a fascinating topic is the approach by using antibody treatment in acute flaccid myelitis. And this is a very novel, fascinating research that has been initiated at Vanderbilt University by Matt Vogt, who is going to present this interesting lecture. And we are going to finish with a very good view of the translational aspect of acute flaccid myelitis and the research that is been developed in the past several years around the topic of virology, immunology, and how that translates to treatment. So with this, we are going to move now to listen the lecture by Dr. Richard Sherman from University of California in San Diego. So thank you very much to all our speakers for the time and effort preparing these lectures, and I appreciate your help for the acute flax myelitis community.